We'll begin. Okay, well, some of you have not been here before, so I have to give my very brief explanation of what this is. So do any of you folks from the Dental Institute, where is it? They took it away.
They took it off.
We have the bridge. What happened to the bridge? Huh?
Presentation.
Oh. Thank you. Okay, so quiz, what is that? Huh? Speak a little louder. Huh? What is it, Courtney? Do you know? That's the Brooklyn Bridge, right. So, you know, why would we have this? Because all of us are in the position of the two gentlemen on the catwalk. We're about to connect Manhattan with Brooklyn. Now, it's very important because once you build a bridge, things are never the same on either side. And so, to many of us, we think that one of the big, huge problems in medicine, health, science today is the fact that two arenas seem to be separated by a huge gap that's even bigger than the East River. And those two dimensions are, one, the explosive developments in basic biological science, bioengineering, mathematics, and so forth. And on the other side is human health and the practice of medicine. To communicate, you have to evolve a certain common kind of language. So the purpose of this course is to help with linguistics, with communication, and primarily in bridge building. So the intent is not to overwhelm you with data and facts, as in a medical school lecture where you have to write everything down and spew it back. It's to excite you about where we are and what the problems are, both from a somewhat clinical perspective and also from a somewhat more basic science perspective. So that's the whole theme of it, and that's the reason of the Brooklyn Bridge. So today's topic of I thought it was periodontitis, but it's periodontitis. So I've learned something already. Is a major problem with an increasing elderly population. I read someplace that half the people over the age of 50 have problems with their gums and develop periodontitis, and they can lose their teeth and demineralize their bone and get all sorts of serious problems with an older population in particular, but not restricted to them, is very serious business. And yet I felt I knew next to nothing about it, and I suspect that many of my colleagues who spend time in other laboratories are in the same sort of a way. So this seemed like an ideal topic to discuss, and particularly because we have experts here at the NIH whose work has focused on different aspects of this. So in my mind, when I was thinking about this, I said, what's the big thing? If bacteria cause this inflammation of the mouth, why don't you just take an antibiotic and get rid of it, like pneumonia? Well, as you probably all know, it doesn't work that way. So why can't it be cured by antibiotics? And what is this biofilm? It's sort of like a buzzword that pops up in connection with different organs and so forth. But what is a biofilm, and what makes it unique? And what does that have to do with the fact that bacteria are not easily eradicated by antibiotics? Now, one of the interesting things, medicine has a way of repeating itself cyclically over long periods of time. And this idea of what used to be called focal infection, and now is increasingly being raised in discussion as to whether this chronic inflammatory condition of the mouth, related to the oral microbiome in part, It also has something to do with other chronic degenerative type diseases. Some people have proposed even having something to do with cancer. Certainly, arthritis has always been a suspect in relationship. But of course, this is not a new idea. It actually goes back to antiquity. And there was a period of time, not quite antiquitous, but in the 20th century of my childhood, when people had sites of chronic infection removed surgically to ameliorate. Maybe it was arthritis, maybe it was almost anything, but a severe degenerative type of disease. So gallbladders were removed, sinuses were drained, mastoids, lots of things, teeth, all to remove this. And then it sort of died down. And then there was a period of autoimmunity. Autoimmune diseases came into their own. And so the issue of focal infection as a factor, but how this could possibly be related was never explained because we didn't know about inflammation in those days, at least as much as known's now. So this has kind of recurred, and we're going to hear about this as to whether periodontitis is a factor in chronic disease of other organs, not just restricted to the mouth. And of course, the mouth is the site of perhaps the first microbiome that a child is exposed to. I'm not sure about that. But at any rate, a unique population of bacteria that far outnumber the number of cells that we have in our body. We're sort of like almost prisoners to it. So at any rate, today's discussion will begin with Niki Moutsopoulos, who all of the first two speakers are in the NIDCR, the National Institute of Dentistry and Craniopharyngeal Research. Niki graduated from dental school in Greece, in Thessaloniki, was trained in periodontics at the University of Maryland, got a Ph.D. when she became interested in immunology as related to periodontal disease. And her main interest and contributions have been in the relationship between the disease and the inflammatory process and how this is related to the microbial population. Niki came to the NIH in 2010, and at present she's a tenure-track clinical investigator. Now, we've published on the website some remarkable articles that she has written with colleagues, and I urge you to look at it. Now, our second speaker is Bob Palmer, who's a microbiologist in that same institute. And his background, he has a PhD in biology from Florida State University, and for much of his scientific work has been interested in the microbiome in relationship to inflammation, environmental microbiology as related to medical microbiology. And his experiences have caused him to work in the Negev in Israel and to deal with samples from the Antarctic and many different parts of the world. He's one of the leading folks around who has for many years studied microbial populations in relationship to this disease. And then our last speaker is Alex Valm. Alex is a ERTA, postdoctoral ERTA fellow in Jennifer Lippincott-Schwartz's lab, where I've got to know him. So Alex got his PhD in pathobiology from Brown, and he has a remarkable track record and interest because he's been interested in analytical and quantitative light microscopy and mathematical computer ways of quantifying what you see, even in living tissues, from the standpoint of microbes. And you're going to hear and see some really extraordinary studies linking the microbial aspect to the disease. So, Niki, would you please begin?
Can I be heard? Am I on? Yes. So thank you, Win, very much. No? Yes. So thank you, Win, very much for including us in your series. It's a pleasure to be here, and thank you for the nice introduction. So what I'll try to do today, and with all of our group together, is to discuss the oral microbiome as it relates to health and disease, and mostly as it relates to periodontitis. But first, I'll start with a little bit of a generic introduction. I know that you all appreciate this, but it's more and more understood that there's more microbial cells on us than our human cells. So as it relates to bacteria, there are 10 times more bacteria than human cells on the human body. Although we're born naked without any bacteria or microbes on us, within a year of life, we become fully colonized, and this complexity of microbes and hosts begins. This complexity becomes even more dramatic when one thinks of genes. So if we think of the contribution of microbial genes on the host versus host genes themselves, it is really, really a more dramatic contribution of microbes versus host that you see on the body. And so this whole theory has come up, and different way of thinking of humans being thought of as a composite of microbial and human cells, and that the human genetic landscape is an aggregate of genes in the human genome and the microbiome together, creating really a superorganism. And so because of this shift in thinking, the Human Microbiome Project was launched. And what came from the Human Microbiome Project initially was first a characterization of what kinds of bugs are on us. And so the people that run the project sampled patients in different sites, and what they saw is that there's distinct ecological niches throughout the human body. And the major ecological niches are the nasal cavity, the oral cavity, the gastrointestinal tract, the urogenital tract, and throughout the skin. And when looking at these distinct ecological niches, it became clear that the oral cavity has a huge contribution. So if we look especially at bacteria, of the percent of different bacteria that are contributed from different sites, the oral cavity is really only second to the GI tract in the diversity of bacteria found. Also, when looking at the different sites, it became very clear that different sites on the body have distinct representation of microbial communities, and that each area really looks very different from the other. And with the oral cavity separating out and having a very distinct microbial colonization pattern and type of communities present. Yeah, I'm pressing the wrong thing every time, it seems. Another thing that became very apparent, and this was very nicely done by the Human Microbiome Project, is that within the oral cavity itself, there are specialized, specific niches, ecological niches. So if one looks at the oral cavity, and I'm sure many of you are not familiar in looking in detail inside one's mouth, one thing that is very special about the oral cavity is that you have hard structures, which are the teeth that are coming out of the body. And so those hard structures are very different colonization niches. There's also soft tissues that are differentially specialized. You have mucosal areas, and if here it's not shown, but one specific mucosal area is within the cheek, which we call the buccal mucosa. There's also mucosal areas that are more keratinized, such as the keratinized gingiva in the roof of the mouth, the hard palate, that are more similar in histology with the skin. And we also have a very special epithelium on the tongue that is specialized to perceive taste, and has a bunch of crisps, which also create a very different environment for bacteria to colonize within the crisps. And so, very smartly so, the people that launched the Human Microbiome Project went and sampled these areas separately. And it was very good that they did so, because when they compared these areas, sorry, I should forget about this part. When they compare these areas, both here looking at bacteria, both in general categories of bacteria called phyla, and also more narrow categories of bacteria, and you'll hear more of these from Rob, called species, what they saw is that the areas of the mouth that are here inside the red box are very different in their microbial general colonization than the stool or the nares. But even within the mouth, different areas have a different representation of large families, phyla, but also a differential representation of species, making it very interesting that within the mouth you have different niches, and that also becomes more and more important as we think of predisposition to disease, because different areas of the oral cavity will be predisposed to different types of conditions as well, in which the microbiome potentially plays a role. Now, when we think of the role of the microbiome in general in health, some of the basic concepts that come to mind is that we have commensal microbiome in health in order to prevent pathogens from invading, to prevent us from infection. Other roles, big roles understood of the microbiome is that they help to shape the immune response, and they help in a lot of physiologic functions of the host, including the providing nutrients or processing of nutrients in the GI tract. When it comes to the oral microbiome, what do we understand of what it does in health and disease? What I can say is that we know a lot more, and we've known for many years, because oral microbiology has had a very rich past, that there are distinct and diverse microbial communities in health in the oral cavity, and that these communities shift in their representation with disease, and I'll show you more of that with periodontal disease. However, we know very little of what the oral microbiome does to regulate physiological functions in the oral cavity and in the body. We know a lot more, and there's been a lot more research done on what different microbes and microbial communities could be doing to trigger disease, both locally and mostly locally, but still there's a lot more to understand in that area as well. And so one way that I conceptualized this and wanted to share with you is that we know now, based on history, on many years of oral microbiology and more recent advances with genomic methods, that we have rich and the communities of microbes that live in the mouth. But we don't really know how these oral microbiome communities shape the immune response or shape physiological functions in the oral cavity. We also know that these oral microbiome or microbiota don't just stay in the mouth. Actually, the mouth can be thought of as a portal to the body. So many of the commensals that could be found in the gastrointestinal tract have to first come through the mouth before they go into the gastrointestinal tract. Our food has to go through the mouth, but we also, as individuals, will swallow about 5 to 6 liters of saliva a day. So there's a lot of introduction of oral bacteria throughout the gastrointestinal tract, but there's also a lot of opportunity within the mouth for systemic translocation. There's a lot of minuscule ulcerations that happen, and a lot of possibility, and we know that even with brushing and eating, we'll have translocation of bacteria systemically, and we don't really know what the introduction of oral bacteria to the system will, what their role will be in systemic health and in the system and physiology of the human body. This becomes increasingly interesting as we think of shifts in microbiome and shifts in community and how they could be triggering local and systemic disease. But for the purposes of our presentation today, I will mostly stay on trying to see how we think of the microbiome triggering local disease, which is periodontal disease. And in fact, as Wynn alluded to, periodontitis is a very common, not oral disease, but is a very common human disease. It's actually one of the most common human inflammatory diseases seen. This is the latest report by the CDC indicating that half the adult population of the US will have some level of periodontal disease. And this becomes a lot more with an aging population. So in people 65 years and older, you will see 70% of the population having a level of periodontal disease. This is increased with environmental factors such as smoking. It's increased in special populations. And it's also, for me, very important to say that severe disease is seen in about 8% of the population. And that's a huge percentage when we're talking about disease that has increased morbidities and increased symptoms. Now, what is periodontal disease? A little bit, just a glimpse of what it looks like. Just to look first at anatomy, teeth sit within what we call the alveolar bone, which is the bone inside the jaws. And this bone is supporting the tooth so it can stay in place. It's connected to the tooth with connective tissue attachments, and there's soft tissues that cover it. And this is what a healthy support would look like. Now, in the presence of periodontal disease, and depending on the level of periodontal disease, you have inflammation in the tissue surrounding the teeth, and you start losing the structures that support your tooth. And so you could lose it mildly, moderately, or severely, and that's how we qualify levels of disease. We do this clinically by measuring loss of structure. We can also visualize it in x-rays. Here in this x-ray, you'll see that the bone looks white because the x-rays cannot go as well through it. But in the areas where bone is lost, you'll have black areas that become larger and larger. And this will show you the level of bone that has been lost. So if we look at periodontitis in a cross-section, what we'll see is that on the tooth surface, there is heavy microbial colonization, and that these communities, as I'll explain to you later, have become dysbiotic microbial communities. You'll see severe inflammation in the tissues that surround the teeth, and you'll see loss of structure and specifically loss of bone around teeth. Now, how does this happen? The way we think this is happening and the way we understand and teach periodontal disease is that the bacterial biofilm, and you'll hear more about the biofilm, which is the communities that live on the tooth surface, will stimulate an exaggerated and chronic inflammatory response. And here in this H&E section, you'll see around the tooth tremendous inflammation on the soft tissue surrounding it. Now, we see this exaggerated response in patients that are susceptible, and we don't really understand exactly what susceptibility means. And this chronic inflammatory response will stimulate tissue destruction, and including the tissue destruction, included in tissue destruction will be the activation of osteoclasts that will resorb the bone around the teeth. Now, as I said, we think of the microbiome as a trigger of disease, and in fact, the microbiome of periodontitis is a dysbiotic microbiome, is a special community that has shifted from health. This is one of the recent studies and one of the first studies that has looked with now modern genomic methods at sequencing the microbiome in health versus disease. And what the study has shown really supports a lot of history in microbiology is that you have periodontitis, periodontal health-related microbiome. You have core species that are found both in health and disease. And what you have in periodontitis is an increase in a lot of species, that you have a lot more species, and also that you have species that are correlated or seen in higher abundance with periodontitis. And this has been understood to a big degree for many decades in the oral microbiology field. And it's just seen now under the lens of sequencing in much better detail. But what has been appreciated by people that had been doing a lot of looking at the periodontal microbiome for many decades is that there are groups of bacteria that are associated more with health or more with disease. And this was the classic way that we had been taught about it, of red and orange clusters of bacteria becoming greater with disease, and a lot of the health-associated species shrinking. But one thing, the way I think about it, because we're not going to really teach names of bacteria for this kind of audience, is that the periodontal microbiome, either in health or in disease, is a microbiome of commensals. And a lot of the species that you'll see in health are the same that you'll see in disease, but just changes in proportion is what you're going to see. So if I put this in a conceptual bubble, the way I think of it is that you have a general growth of amount of bacteria. So the load and the burden on the body is going to be much greater of bacteria. And you'll have a lot of the same bubbles and a lot of the same bacteria that are going to be both here in health and here in disease, but just with shifting amounts and a greater number of species in general detected with periodontitis. So the key findings that I would report of the periodontitis microbiome is that there's an increased bacterial burden, that there are increased species detected, although many of these are also found in different patients in health. There's communities that are richer and diverser as you go into periodontitis, and that there are species that we discussed as periodontitis associated that are overrepresented in the disease. Now, the key question is, does the periodontitis microbiome trigger disease in the oral cavity, but also systemically? I will diverge just for a minute, although we have no way to know exactly what the oral microbiome does systemically, but I know that this becomes very interesting to the broader community of medicine and biology, that we have to systemic translocation in general of oral microbes, but this becomes a lot more profound in the presence of periodontitis. So as I just showed you, in periodontitis, you have a greater load of bacteria, so there's a lot more of a potential of microbes to be either swallowed or translocated, but also in periodontitis, you will have ulceration of all of these tissues that become inflamed, and that ulcerated area can become a lot easier a portal for systemic translocation. So what people have found up to now is that you'll detect tooth-adhered bacteria, especially in periodontitis, in different parts of the body. And so people have seen in atheromatic plaques of patients undergoing surgery that there is detection of periodontal microbes. When people have looked recently with genomic methods at the placental microbiome, it seems to resemble most closely the oral microbiome compared to any other type of microbiome in the body. In rheumatoid arthritis, and these links of trying to think of what diseases are associated, we find there's been many reports of periodontal microbes in synovial fluid, but also patients with rheumatoid arthritis will have increased titers to periodontal associated microbes such as P. gingivalis. Also in rheumatoid arthritis patients, there's increased presence of oral microbes in the gut. And one other interesting finding that's been emerging more and more is that an oral microbe, which is a tooth-adhered microbe, again, mostly Fusobacterium nucleatum, is mostly associated with colon cancer. And there's a lot of studies now trying to look at mechanisms of how Fusobacterium nucleatum could perpetuate or trigger carcinogenesis. Now, this slide is not to say that we know in any way that these microbes are triggering the systemic disease, but they're very interesting findings, and that could start clicking our mind into thinking of what the role of these microbes systemically could be. And as we have the ability to look at their role in the mouth, this might educate our understanding of what they could be doing systemically as well. Now, for periodontitis, coming back to periodontitis though, is the microbiome a trigger or a consequence? We know for sure that the microbiome in periodontitis has a level of triggering disease. And we know this mostly based on clinical studies. A lot of what we know for periodontitis conclusively comes from human data and from studies that have happened decades ago. So you know from just going to the dentist that preventive care for gingivitis and periodontitis is routine cleanings. In those cleanings, what we do is a gross mechanical removal of plaque. And that's how we try to either prevent disease, arrest disease, slow down disease, and also in aggressive forms of disease, we use antibiotics. So we trigger this, our treatment based on a lot of data that has come from many, many decades ago. But there's many things that we don't know. We don't know if the microbiome is an initial trigger, or if it really, or this biotic microbiome establishes itself in a favorable inflammatory environment and then will just become a secondary perpetuant. We don't know if patients that are susceptible would just respond with exaggerated responses to any microbiome, to any microbial stimulus. And we also don't know if host susceptibility just predisposes you to becoming colonized with specific bacteria either. And so there's a lot more for us to understand of how this imbalance between microbiome and host occurs. Now, when we think in general of how microbiome establishes itself on the human body, I would say that there's host-dependent factors and external factors that play a big role. So the host genotype it plays a role in what kind of microbiome establishes, and we know that there's differences in individuals of what microbiome they carry, even in health. The presence of health and disease will shape the microbiome. The immune response will shape the microbiome. And all these things have to do with the host themselves. But there's a lot of external factors ranging from where we live, what we eat, parts of our lifestyle, if we take medications, the environment that we're in. But My group has been interested mostly in the host genotype and how one's genetic susceptibility could predispose you to either a differential immune response or a differential microbial colonization. And for the purposes of this study, I'm focusing mostly on the microbiome part of our work. And again, as Wynn said, we don't, for this type of presentation, we try to keep our data to the minimal, but we're just trying to introduce work that may start shifting concepts in our field. So the way we've been approaching some of the problems is, because of the unique setting of the NIH hospital, is to look at patients with single gene disruptions, and particularly with single gene disruptions in human immunity. And while looking both at the literature and patients that come to the hospital, we're trying to categorize what types of immune defects would predispose you to what type of oral conditions in the oral cavity. And so when it comes to periodontitis, based on many previous observations, and also our own in the NIH clinic, we know that patients that have issues with the immune cell called neutrophil will have aggressive presentations of periodontal disease. Just a small introduction of what a neutrophil is, and this is by no means in detail, is that in general we think of neutrophils as innate immune cells that play a big role in infection control. And these are some of the first cells in the human body that will come in and try to control particularly bacterial infections. And they have many ways of doing that. They can phagocytose, they can degranulate, they spit out nets. And so patients that have neutrophil defects in general were thought of as patients that are susceptible to infection. We've been studying closely patients with a syndrome called leukocyte adhesion deficiency. These are patients that have neutrophils, but their neutrophils have a mutation that does not allow their neutrophils to come within the tissue and provide support in the tissue level. These patients have very severe periodontitis and recurrent oral ulcers in the oral cavity, as well as life-threatening infections and colitis later in life. This is an example of one of our patients at the NIH Center, and what I've drawn for you here is the top line representing approximately where bone should have been. Bone levels should have been in this patient at such a young age. And the lower level shows you where the bone has gone. And so it's showing you that there's a dramatic loss of support around the teeth in this disease. So we have very severe, extreme clinical phenotypes in these patients. When we look at their microbiome colonization and their immune response in the gross level, what we see is that on the tooth surface, you have a high burden of bacteria, but minimal bacteria within the tissue, or really undetected bacteria. So there seems to be a microbial stimulus that could be on the tooth, but not within the tissues. Within the tissues, we see a tremendous inflammatory response. And here you'll see an H&E stain. This is an extracted tooth. And all these tissues that are surrounded have tremendous inflammation. But what we don't know is what the interplay here is, if the microbiome triggers this immune response, or if the immune response just creates a very conducive environment for microbiome to grow. And many of these things, as you'll see, are very hard to dissect in humans, but we can get clues from some of our patients. And what we did is to see, is this microbiome on these patients unique in any way? And indeed it was. When we looked at the microbiome that develops on these patients compared to health, it separates out very much. It also separates out from the microbiome that one would see from classic periodontitis patients. This is a heat map of different species of bacteria detected in health versus LAD, and you'll see that these two groups are very different, and the biggest difference is that the LAD patients lack a lot of the health-associated bacteria. So a lot of the healthy commensals are lost, and what we did have is a few distinct species that are overrepresented. But what we don't know is, does the immune defect predispose to an altered microbiome colonization? Does the altered immune response because of the defect then just allow for different microbes to colonize? And is this altered microbiome one way or another an initial or a secondary disease trigger? So one more thing that we did to look into that is try to conceptualize how microbes in this setting could be triggering disease. So when we looked at the tooth surface, we had seen with gram stains that there's a lot of bacteria on the tooth, but not in in the tissue surrounding. But when we did more extensive studies, we realized that parts of bacteria, such as LPS, can shed from the microbes and go within the tissue and actually surround the inflammatory cells, giving us an idea that parts of bacteria could be stimulating this local response. To look at this a little bit deeper, we wanted to ask the question, is the microbiome of LAD triggering a higher inflammatory disease or a different inflammatory disease compared to microbiome from healthy patients? So we took the biofilm from health and biofilm from LAD patients, and we stimulated either in vitro immune cells or animals in vivo and looked at the immune response. And I'll just tell you as a general message of what we saw is that the LAD microbiome had an ability to stimulate increased inflammatory responses, and that these inflammatory responses had a special type. They were predominated by IL-23 and IL-17 specific cytokines and their pathways. And those were actually the cytokines and responses that we found within the tissues of LAD patients when we looked there. The reason that became interesting to us, and I think it's worth, and I thought was more worth mentioning, is that this IL-17 and 23 response made sense to be upregulated. This isn't normally a response that causes neutrophils. So in patients that have neutrophil problems or neutrophil recruitment defects, it makes sense that you would upregulate a response that is just calling some of these cells to come back in. But the other reason this became very interesting to us is that this response, when exaggerated, is a response that will trigger tissue degradation and specifically bone loss. So IL-17 has a direct way to activate osteoclasts and make them resorb bone. So this started opening the road for us to start thinking of specific immune pathways that we could be targeting when thinking of targeting this disease that is not responsive to standard of care. But going back to our microbiome thoughts and trying to opening the door, because our lab is more of an immunology lab, so we think of more targeting the immune response. But to open this up, because this is more of an oral microbiome setting, when we think in general of how we want to target periodontitis and specifically LAD periodontitis, we think that it's the host susceptibility that sets you up either to be colonized with a dysbiotic microbiome or to be responding with a more destructive inflammation or both, and that there could be a two-way street where the microbiome triggers inflammation but also that inflammation will set you up for an alternate microbiome. And so when we think of what should we be targeting here and what should we be targeting in general in periodontitis, one easy way out would be to just reverse the host susceptibility. And that is actually feasible in monogenic patients because these patients get bone marrow transplants. Now there's a lot more talk and effort in trying to get to have genetic therapy, gene therapy approaches. And in fact, the patients with these types of defects, when they get bone marrow transplant, their disease is reversed. Their periodontal disease either doesn't occur if it hasn't happened before or will become arrested. But this would not be an option in a multi-agenic kind of scenario such as periodontitis. And then we want to think of, are there targeted ways to either target inflammation or to target microbial colonization? And so as I told you, with inflammation, we're thinking a lot more about targeting specific pathways, and we've looked a lot more of what pathways are specific to our diseases in order to target them. But how do we start thinking of targeting the microbiome in a more targeted way and not in a gross, just removing mechanically whatever we can type of way? And what I want to propose, and I think this is a very good segue in what you'll see in the next presentations, is that we need to understand more about microbial communities before we can target them into becoming dysbiotic. And so if one thinks of dysbiosis as a continuum in which health-associated communities will progress into a disease state, it's important to know who's there in a health-associated community, and we know a lot of that. But it's very important to also know how members of the community interact. What of these interactions are very important to keep these communities in a healthy state? And then what is a breaking point? What is an interaction that if you would break it, or a microbe that if you introduce it in a greater abundance will become a keystone or become a transition that leads you into a more dysbiotic situation. And once you go into dysbiosis, what are the key microbiomes, the microbes that will sustain a dysbiotic environment? What are the relationships there that make that stable and harder to attack? And when it comes to the inflammatory response, are there parts of bacteria that are the most important in driving most destructive inflammatory responses? And so what I want to propose is that the more we know about biofilm formation and biofilm transition to disease, the more we have a hope in trying to start to think about targeted interventions into these microbial communities in periodontitis of any sort. And I think that what you'll see next today is a lot more information of how we understand the dental biofilm, how we can visualize it, and what more we need to know of its transition into a disease state to start to be able to educate therapeutic interventions in the future. I will leave you with a large acknowledgement slide. We are working with many people on campus and extramurally to try to have a global kind of approach in how we target our questions. And thank you very much for your attention.
Because people are watching and they like to hear the question.
I come from the clinical side of the bridge and want to ask a question. We're taught that the elderly individuals have recession of the gums. Is that a degenerative disease or is that an inflammatory disease related to periodontitis?
So
how do they interact?
So I'll give you some level of response, and then there's more people that can also measure in here because we have a good periodontitis to other ratio in the room. So gingival recession could be many things. When you lose bone, then many times the soft tissue will come with it. So it could be a result of an inflammatory disease. Many times you'll have gingival recession also because of overbrushing, and it becomes because of too much of a triggering response that is not always periodontitis related. So it could be either.
So, Niki, is periodontal disease a disease of other mammals?
So I know for sure that dogs have naturally occurring periodontitis. And also that there's vets that are specialized in cleaning teeth and that have that's why the dog model had been also a naturally occurring, natural progression model. My understanding is that there's types of monkeys that will have naturally occurring periodontitis, and they're not all of them. Now, interestingly, in mice, you have a progression of bone loss with age I don't know that we could call periodontitis, though. We just know that as mice age, you'll have more alveolar bone loss, and that's also been used as a model as well.
So, in the case of healthy a child and an adult, do you see the microbiome community members being pretty much static, or there is a diversity in the microbiome community over time?
So my understanding is, and Martha can chime in, is that there's a lot of studies now going on and trying to look at the progression of the microbiome with age. The early studies speak to increased diversity with age, but there's a lot now that is going on, and we don't have results from a lot of these studies that is looking with genomic methods to look within the same individual or within families how the microbiome progresses with time. So hopefully we'll know more.
Please, no questions without microscope.
So interestingly enough, in some of the work that we're funding through NIDCR and the extramural community, there's a suggestion, although it's only in abstract form, that in fact the oral microbiome is more static with aging versus the gut. Whether that's going to hold true or not is fascinating, and we're still waiting. So there's a lot of funding in the area. So just to clarify that.
So do we know, or is Bob going to discuss how nutrition influences microbiome?
I don't think Rob's going
Nutrition
to discuss that.
influences the microbiome.
I don't know that Rob's going to discuss it, and I don't know that we have, again, I don't know if there's funding on that area as well, but I don't know that we have very solid data of differences in microbiome with nutrition in the mouth. There's a lot more in the GI tract on that end.
I've enjoyed your talk so far. Can you explain to me, perhaps I have a misconception, but it's my understanding that you can have just one tooth out of many that are affected by periodontal disease, and you talked about both local and distal infections. So could you explain how that could be? And then could you also comment on the response when you have a dental implant and what happens, and whether or not, in fact, with people with severe disease, there ought to be preventive use of implants, because my understanding that the periodontitis goes away with an implant.
So that twofold question. So one is localized periodontitis, and I didn't touch on that, but in fact, in the same mouth, you can have areas that are with more or less severe disease, and that could be one of many things. Many times when we have local disease, it has to do, something's wrong, is that okay? So it has to do with many times local etiologic factors. So we see clinically that an area where somebody will have a restoration or in areas where microbiome can colonize in a more excessive way, that those areas might have a more severe disease within the same individual rather than others. That was the first part of the question. So that explains at least some part of the local disease. But then your second question was implants, and if it goes away. That's not exactly true. Patients that have severe and aggressive, especially forms of periodontitis, will have higher risks of developing what we call peri-implantitis, which is periodontitis around implants, which is again the idea of a microbiome stimulated loss of structure around the implants as well.
So we did touch about biofilm, and my question is, so after the mechanical removal of this bacteria during the dental visit, how fast it repopulates?
That's a great question. So that's a great question. I can tell you clinically what we do, but I think that there's a lot more studies that need to happen of trying to see, because we can look at how clinically the reason how we spread out visits and cleanings has to do with what we've seen clinically of where does the inflammation start again. And so depending on anywhere from 3 to 6 months is where we usually spread our visits, because we realize that around that time you'll have again microbiome stimulated responses. But we're still lacking in trying to understand how does it redevelop, how fast it redevelops, how fast does it take to become a biofilm? There's a lot of things that we don't know yet, but the beauty of the oral cavity is that that's one of the few areas in the body that you could study all these things. And you'll see, Rob, I'm not going to give all of his stuff away, that he has tools, or they've been working on tools of trying to look at colonization, time for colonization, and that I think what we were trying to do as a group is also pitch the idea that understanding biofilm formation, understanding microbiome, understanding responses to microbiome can be done in the oral cavity in humans better than can be done in most other sites that are internal. Maybe skin is the only other place.
I think we have one more, Bob.
Have you noticed large differences regionally or internationally in the oral microbiome?
So I think most of the data that we have is from the HMP. I've only seen one, and that's most of the bigger study that people have been binding. And so I don't know that we can answer your question, especially with the genomic stuff internationally at this point. It would be very interesting in some of these in different populations around the world that people are doing.
Okay, I think we'll have time for more questions later. Thank you very much, Niki. That was excellent. So, Bob?
Great. Well, good afternoon, everybody. Wynn, it's a great honor to be part of this lecture series that you've so faithfully and magnificently shepherded for so many years. My goal here today is to give you a whirlwind tour of microbial bacterial taxonomy, microbial ecology, and biofilm formation as is relevant to the oral cavity. The story starts here. This gentleman, contrary to popular belief, did not invent the microscope, but he produced the best lenses out there. And at that time, brushing and flossing wasn't very popular, and he always had lots of this white stuff on his teeth to scrape off and look at in his microscope. And he saw this wonderful collection of these different shaped organisms, which we later realized are bacteria. This was the first study of bacterial biofilm. So let's fast forward about 300 years. Microbial ecologists were having an existential crisis called the Great Plate Count Anomaly. And organisms in nature were enumerated back then by simply taking a sample, spreading it out on bacteriological culture medium, allowing colonies to develop there, and then counting the number of colonies on the plate, and translates to a number of bacteria per unit volume. At the same time, nucleic acid stains were being brought in, and so investigators could see all these organisms in the sample. And so if you take that stain sample and spread it onto a filter disc, and look at that filter disc in the microscope, and you can also count the number of bacteria on the filter disc, which gives you an alternate estimate of the number of bacteria per unit volume. The crisis was that the number of bacteria counted in the microscope was always much, much greater than the number of bacteria seen on the plates. And so after many experiments involving different kinds of culture medium and different ways of counting things, an estimate was arrived at that roughly 1% of bacteria in nature were actually cultivated. At that same time, this gentleman was working on the molecular end of things. Does anyone know who this guy is? This is a picture from his obituary that appeared in Science a couple years ago, and he is? That's right. Carl Woese would change the face of taxonomy, biological taxonomy, in a very profound way. Woese realized that ribosomes are molecular clocks. They're universal, they're present in all living organisms, they're made up of conserved and variable regions which allow them to evolve, the sequence to evolve faster than whole genome sequences, but slower than protein sequences. And so these small subunits of the ribosome, referred to as the 16S from bacteria and the 18S from nucleated organisms, these were ideal size for separation and sequencing, especially when you're doing it like this. Now, this is Woese's seminal contribution, a paper in PNAS that consists of a single table. Woese took these groups of organisms, nucleated cells, like the yeast, duckweed, L cell, which is, I believe, some sort of tumor cell from mice, and then this bunch of organisms, which were generally, they're anucleate, single-celled things that were just referred to as bacteria. And he examined the relatedness of the 16 and 18S sequences among these organisms by using what he called an association coefficient, which is basically a fractional number with one being identity. And when he looked at this, the nucleated organisms were were more similar to each other than they were to all of the other organisms. This group, this group of bacteria was more similar to amongst its own within itself than to the nucleated organisms or to this group. Now, this was really the important finding, because these two groups of organisms here were really considered one at the time. And Woese was the person who demonstrated then that in biology there are three primary kingdoms: the eukaryotes, the eubacteria, and then what came to be known as the archaebacteria. Now, those of you with good eyes will see that there is a line missing here, and that's the chloroplast isolated from duckweed. And this chloroplast is more similar to eubacteria than it is to the organism from which it was obtained. And this, of course, is the underpinning of the endosymbiotic theory of organelle acquisition by nucleated cells. So by translating these association coefficients into lines where the length of the line represents the evolutionary distance from its nearest neighbor, you can make these wonderful trees. And this is the tree that I call the tree of life from the perspective of cell biologists. So you see all of these wonderful organisms, the insects, the reptiles, fungi, and all springing from some original single nucleated cell, and then relegated to the basement here are bacteria, eubacteria, and archaebacteria. But from the perspective of a microbiologist, and what I would call that more accurate perspective, there are many, many more families and species of bacteria and of archaea than there are of nucleated organisms. Now, all of this microbiome stuff actually started in environmental microbiology well before the Human Microbiome Project. And environmental microbiologists realized that you could use these 16S sequences to characterize entire populations. And this is the standard scheme for how that's done. You take a sample, you extract the nucleic acids, you can amplify specifically these ribosomal RNA sequences. We're working now with genes rather than actual ribosomes. Or you can directly go from these extracted nucleic acids to a catalog of ribosomal DNA, the genes of these that are code for these ribosomes. These sequences form a catalog of organisms in the sample. And by comparing that catalog to a catalog composed of sequences from known organisms, you can get a picture of what organisms are there, the so-called microbiome. Now, microbial ecologists suddenly realized that this great plate count anomaly is solved through this process of RNA-based taxonomy. That is, these organisms that don't grow on these numerous kinds of culture media can now be detected and classified because of their presence of the presence of the RNA sequences. But that threw bacterial taxonomy for a bit of a loop, because in order to have a valid bacteriological species, you require a pure culture of that organism is required, so you can investigate its physiology and to nowadays sequence the entire genome of that organism. But because you have these uncultivable or not yet cultivated organisms, you have a molecular species that's defined solely by sequence data. These are referred to as operational taxonomic units, various derivatives of that term. But the important thing to realize here is that all valid bacteriological species are molecular species because they have an OTU that represents that organism. But not all OTUs are in turn valid bacteriological species because no pure culture, in many cases, no pure culture of this organism exists. But yet we recognize these to be species level, indicator of a species level organism in the sample. Now, what does this mean for the oral cavity? Here are the major OTUs from supragingival plaque, the toothed surface, and subgingival plaque, the area below the gum line. It's associated with periodontal disease. Now, if you just count the OTUs, in supragingival plaque, there are 52 major OTUs, and less than 50% of those organisms were yet to be cultured. Now, these data are a little old, so that number is now getting progressively smaller. If you look at subgingival plaque, you've got about an order of magnitude more diversity there, about 350 OTUs. But again, 50% of these organisms have already been cultured at that time, 10 years ago. Now, If you look at the entire oral cavity, including the mucosal surfaces, there's roughly 700 major OTUs, and only about 40%, actually 40% of those, in fact, have been cultured. So we know an awful lot about these communities by culturing, and you can see that there's easily more than an order of magnitude of organisms cultivated from these communities than there have been from natural environments outside the human body. So let's look at this a little bit closer. We are going to have some species names here, Niki. So let's think about these in terms of the way that you see these often presented in papers, which is by phyla. And so here are the Firmicutes, and this phyla has two arms. There are the bacilli, which are primarily streptococci. And these streptococci, if you don't have to read all of these names, but they're very similar names from subgingival to supragingival environments. So they're really the sort of same types of organisms there. But now there's also another arm, the clostridial arm, and this arm varies quite significantly between subgingival plaque and supragingival plaque. Now, there are a couple other phyla here, the actinobacteria. There's some marginal differences there between supragingival and subgingival. Fusobacteria, not a lot of difference at all between the two sites. Proteobacteria, these are your sort of E. coli type organisms, aerobic and heterotrophic. Not a big difference there. The Bacteroidetes, which contains organisms like Porphyromonas and Prevotella. These are major organisms associated with periodontal disease. But again, these organisms are found in supragingival plaque as well as subgingival plaque. And so there's really not a tremendous difference in the composition of these communities dependent on the site. Now, there are two groups here, the two phyla here, the Synergistides and the Spirochetes, which are unique really to subgingival plaque. The Spirochetes consist of treponemes, and the Synergistides had no cultivated representatives until about 2 years ago. And then this final organism here, called TM7, this organism was one of these things that was known only as a molecular species until very recently, in the past year, when it has been sort of cultivated. There is a co-culture of this organism together with an actinomyces as a helper organism. So this still doesn't give us quite enough to give it a valid bacteriological species name, but we're almost there. So what does this community structure mean in terms of periodontal disease? Well, this is a wonderful study in which the subgingival pockets of 29 healthy subjects were sampled, and then 29 subjects with chronic periodontitis, severe periodontitis were sampled. But within those people, there were some shallow pockets where not much bone loss or attachment loss occurred, and these they called healthy sites. I called them about to be diseased sites. And then the deep pockets, which are the truly diseased sites. And if you look at the microbiomes here represented by each of these dots, there's 29 dots for each cohort of samples here, you see that these groups of samples cluster in with an amazing degree of integrity. And you also see that each of those these 29 dots don't overlap each other very much. And so that shows that each of us is a unique individual. But there is this difference in the microbiota associated with the clinical definition of disease. Now, to look a little bit closer at that, here we see arrayed here all of the organisms that were detected, OTUs, in this study, displayed as the percentage of the total of the population in these three sample cohorts. And what you can see here is that you have a group of organisms that are prominent in disease. That means the red bar, the disease sites, is higher, greater proportion of these organisms in disease than in health, but the organisms are in fact there in health. And conversely, you have organisms that are more prominent in health than they are in disease. And really what to me was an amazing point in this study was that these patient healthy sites, the about to be disease sites, almost exclusively fell in between the severely diseased sites and the healthy sites. Now, for me, the interesting question is, how do these communities assemble? And one way that's unique to the oral cavity, or very close to unique to the oral cavity, revolves around a process of cell-cell recognition called coaggregation. It's been known for decades that if you grow oral isolates up in pure cultures, there's organism 1 and organism 2, and you mix them together, if they recognize each other by coaggregation, they flocculate. And this flocculation is sometimes so strong and the flock so large that the coaggregates settle to the bottom of the tube in a matter of seconds. Now, you can reverse this coaggregation and obtain a homogeneous suspension again by adding sugar or protease to the mixture. And this then tells you that this is a lectin-like interaction in which a cell surface protein adhesion recognizes a complementary cell surface carbohydrate on the coaggregating partner. Now, through examining many oral isolates from all sorts of culture collections, these sorts of diagrams can be built up that recapitulate the development of dental plaque based on these adhesion receptor interactions. And they begin with streptococci, which have adhesins that recognize salivary proteins, which are in turn adsorbed onto the tooth surface. So this is the first layer. And then these other organisms come into the community based on their ability to recognize the streptococci or the ones that are attached to the streptococci and so on and so forth. And the ones that come in last are invariably these organisms that are associated with periodontal disease, like the treponemes and porphyromonads. Now, this is a wonderful model that appears in dental textbooks everywhere. But until a couple of years ago, there was always a question about, well, does this actually occur in vivo? I mean, it's a nice observation, but does this happen in real life? Well, we set out to do that, to answer this question. And so the big question was, how can we assess the relevance in vivo? And Van Leeuwenhoek told us how to do that. You do it with microscopy. And the reason is because microscopy is the only way to obtain spatial and temporal data on these developing biofilms. We set up a clinical protocol to look at this. Healthy volunteer wears 2 stents, and each of those stents carries a couple of enamel chips that were cut from extracted third molars. And we take those chips out after 4 hours and 8 hours of wear. And we stain them with antibodies against these adhesins and receptors, and we look at the biofilm with laser confocal microscopy. Now, here's a little model that shows the coaggregation interactions about which the most is known. So you have streptococci here, and they bear this receptor polysaccharide, the carbohydrate component on their cell surface. There are 2 flavors, G and GN. That's not really important, but there are a group of streptococci that bear adhesins, protein adhesins on their cell surface that recognize this GN receptor polysaccharide. In turn, there are other organisms like actinomyces that have these organelles, fimbriae, little hairy projections that stick out from their cell surface that are the protein adhesins, and these adhesins in fact recognize both of these receptor polysaccharides, both flavors of the receptor polysaccharide. Now let's go looking for these interactions in nature. So we make an antibody against, whole cell antibody against this organism here. That's a pretty decent antibody. It recognizes most of the streptococci that has these adhesins. And then we make an antibody against the purified polysaccharide from this streptococcus. We go in and we look and see what we can find. This is early plaque development here. You can see some single cells here, but you see this nice little cluster here of a single receptor polysaccharide bearing cell in close association with cells stained with this antibody here. Now that looks nice, but my bar is set really high for this kind of stuff. And this antibody, it doesn't recognize all of the receptor or all of the adhesion-bearing streptococci, and there is some cross-reactivity because it's made against whole cells. There is some cross-reactivity with other streptococci, not a lot, but it happens. And so we'd like to get a little better defined system here. So to do that, we make an antibody against the protein adhesin here, the fimbriae on the actinomyces. And now we go in, we look with these antibodies, And we see here these piles of receptor polysaccharide-bearing cells, and here you can see one, maybe two cells that are stained with the antibody to the fimbriae, and here a single cell pressed up against one or two receptor-bearing cells. And so this then is very good evidence, and that these adhesion receptor interactions are very important for establishing the oral biofilm on the tooth surface. So let's review a little bit what we know about oral biofilms from microscopy. Because the oral biofilms are so accessible, they're one of the first things to be looked at with electron microscopy. And back in the late 80s, the same sort of model system that we used was used to produce samples for EM. Here you see early at 4 hours. This is when you can just begin to find bacteria on the tooth surface with EM. Here's a little colony or two cells there. Are they the same cell? Did they arise by division? Are they two separate cells? Well, we don't know. Here you see after 8 hours, these colonies can increase in size. There's some morphological diversity here if you look here carefully, but we don't really have a functional understanding of what these colonies are. Now, with targeted antibodies, functional approaches to understanding the ecosystem, we know that these early communities are, from the very beginning, multi-species communities of different cells that seem to arise through interactions of these receptor and adhesion receptor interactions. Now, after 12 hours, you see these sheets of biofilm developing here. And again, if you look at the leading edge of one of these sheets, you can see this morphological diversity, but you don't really know what those things are. But now, with these more targeted approaches, we know for a fact that these biofilms are very complex, highly interactive collections, multi-species collections. And let me now just review what I hope I've made clear to you. We know an awful lot about the oral environment through culturing of bacteria, classical bacteriology. What molecular taxonomy has done is to identify some of these things that haven't been cultured, but its big advantage is it rapidly provides complete community descriptions without having to do any culturing whatsoever. I've shown you, I hope, that the microflora of disease sites clearly is different from that of healthy sites, but the difference is that of proportions of organisms, not specific organisms. So there is not a pathogen, the community is a pathogen. I also hope I've been able to convince you that these oral biofilms are, from the very nascent stages, multi-species communities, and that cell-cell interactions play a role in the development of these biofilms. And, you know, I do a lot of microscopy, and basically you might have noticed I'm working with maybe 2, 3, 4 colors at a time. And I've always wished, you know, it'd be really great to be able to look at these things with more than a handful of colors. And our next speaker is going to tell you all about that. But first, I'd like to acknowledge 2 giants in the area of coaggregation with whom I've had the pleasure of working at the NIDCR, and this guy who started it all. Thanks very much for your attention. It's absolutely clear. Oh. Can you see how it's attached? I can't see the gravity.
Thank you. Okay, so I also want to start by thanking Wynn for giving me this tremendous opportunity here. I've been, I guess, a student in this class for a couple of years, and now it's my pleasure to be able to give back to this class. Okay, aha, he turned it up. All right, so I also start my talk off with this same graphic that Rob started his talk with. and I wanted to tell a little story actually about this. So I had also the great pleasure of being able to read one of the first of the English translations of this letter that Van Leeuwenhoek wrote to the Royal Society when I was in grad school at Brown. And when I saw this graphic in that form, I actually was struck almost immediately with two ideas. One was something that other people have had before, which is that the very first direct observation of bacteria revealed that the human body is filled with bacteria. Obviously, the motivation behind the Human Microbiome Project. But the other thought that I had was that also this very first observation revealed that microbes live in very complex communities. They don't live in nature as single species, which is how we've been studying them almost ever since. So that was the Human Microbiome Project circa 1700. Now in 2016, we study the human microbiome not primarily through microscopy, but through molecular methods by sequencing the DNA that we collect from different parts of the human body. So we can do this in a very high throughput way thanks to tremendous advances in DNA sequencing technology as well as in the bioinformatics approaches that we've developed to analyze this data. So this allows us to survey the human microbiome in terms of, on the one hand, what organisms are present in the human microbiome, as well as what functions these organisms have. And we do that by identifying genes that are present in these organisms. And so we collect all this data, this sort of list of parts, this list of organisms and genes that are present in the human microbiome. And the next step is to try to distill function from this parts list. And the way that that's been done is by a mathematical approach called network inference. So this graph here shows you, it's a summary of all of the data from the Human Microbiome Project. Each of these dots here represents a kind of bacteria. I think they're mostly here as families, so the family level of bacteria. The color of the dot here represents where in the human body this bacteria is primarily found. And then there are lines that are very hard to see here that connect some of the dots. A green line between two dots means that there's a positive correlation between the two kinds of bacteria, that they're found together. And a red line means that there's a negative correlation, meaning that they're often or they're never found together in the same person or the same sample. And so we build a network like this that gives us some idea of the structure of the human microbiome. And we do see that there is some structure on this kind of scale, so that these organisms in these red organisms all come from the mouth, and they tend to have relationships with each other. These gray organisms here are from stool or from the gut. They cluster here, organisms here from the urogenital tract and the skin. And so these clusters form where organisms in some part of the body tend to have relationships, and they're separate from other parts of the body. But then we can ask, given that bacteria are orders of magnitude smaller than a human, is this a relevant spatial scale for trying to distill function from this data? Obviously, no. well, it is, but there's more that we can learn from finer scales, right? So there are some hypotheses that exist regarding the micron scale structure of the human microbiome. In the gut, it's hypothesized that it's very important that in states of health, that the bacteria of the gut are separated from the epithelial cells in the gut by a mucus layer, and that removal of this mucus layer contributes to diseases of the gut. As well, in the skin, it's hypothesized that the microbes live in the most superficial layers of the epidermis, and that they can also be found within skin appendages like hair follicles, and that this is actually very important for interacting with cells of the immune system. Rob also showed a slide just like this. My point for this slide is to sort of maybe hammer home something that Rob did say, which is that the micron level spatial structure of the human oral microbiome might be especially important, because in this hypothesis, this model is a hypothesis. Like Rob said, it has not been directly observed. It's been distilled from piecemeal molecular data. But this hypothesis posits that the organisms that are associated with periodontitis are structured, that this whole community is structured so that the organisms associated with disease are located in one part of this structure. Organisms associated with health are in a different part of this structure. So this sort of demands that we study the oral microbiome from using microscopy on a micron level scale. And Rob showed images using antibodies, another method for being able to visualize microbes. That's particularly important in the age of DNA sequencing, I argue, is fluorescence in situ hybridization, so FISH. So with this approach, if you know the sequence of a target molecule in your organism, for example, the 16S rRNA, you can design an oligonucleotide probe, a single-stranded probe that will bind specifically to your organism of interest and not to other organisms that you know or suspect might be present in the community. You can conjugate this nucleotide or this polynucleotide to a fluorescent reporter, and you can use different reporters to be able to differentially identify different species present in a sample. So through microscopy, we have been able to sort of verify some of these hypotheses. For example, that yes, in health, the bacteria, the microbiome, the microbiota of the gut are in fact separated by some distance from the epithelial cells of the gut, and that in the skin, bacteria and fungi live together in biofilms on hair follicles deep within the epidermis. Now, a limitation, though, of fluorescence in situ hybridization, and in fact of any kind of fluorescence microscopy, is that, as Rob pointed out, it's easy to distinguish 2 or 3 different colors, fluorescent reporters, but it's almost impossible to do that for more than just a handful of fluorescent dyes. And that's due to the photophysical properties of fluorescent reporters, or at least the ones that are appropriate for biological imaging. And namely, that's that they have these sort of broad emission spectra. And so when we use filters in our microscopes, we can't distinguish more than 2 or 3 or 4 different colors usually. So to summarize, the differences between a molecular sequencing assay versus an imaging assay, DNA sequencing has some strengths, which are that it has a high breadth, meaning that we get lists of almost, in theory, we can get an entire list of all the organisms that are present in a sample. And we don't need to know anything about that sample before we sequence it. This is in contrast to imaging, where we can only identify a few organisms at any single time. And we have to already know what we think is there, and we have to know the sequence of their 16S ribosomal RNA before we can even attempt to try and visualize them. On the other hand, DNA sequencing has a relatively low detection limit and also gives us very little information on spatial distribution in contrast to imaging. So my work has been to try and develop a technology that could bridge these 2 types of approaches, and that could draw on the strengths of both DNA sequencing and imaging. And so this technology that I've been developing as a grad student, and now again here at the NIH as a postdoc, has 2 parts to it. So the first part is that we use fluorescence spectral imaging. So these are commercially available microscopes that, instead of using fluorescence filters in the light path to discriminate different fluorophores, they have a sort of crude spectrometer attached in front of the detector. So we sample the entire emitted spectrum at every pixel in the image. Then we use a computational approach borrowed from satellite imaging, actually, called linear unmixing, where we can, if we have some unknown spectrum that we've measured in our image, and if we know what the fluorescent spectra are of the fluorophores that we used to label our microbes, we can use a computational approach to find the best fit of our observed spectrum to our known fluorophores. So we can identify what fluorophores and thus what organisms are present and where they are in our image. So that's the first part of the new technique. The second part is combinatorial labeling. So instead of labeling bacteria with only one fluorophore, we actually label our bacteria with combinations of fluorophores. And so to start off with, we just did binary combinations. And so I'm showing you here the power of combinations. With only 8 fluorophores, there exist actually 28 different binary combinations. So we end up labeling our microbes with fluorescent signatures. And so taken together, we named our technique ClassyFISH for combinatorial labeling and spectral imaging. And so we first set out to prove in principle that we could actually identify bacteria labeled with this many different colors. So we just took a laboratory stock of E. coli and solutions of 8 different fluorophores. So these are fish probes that all bind to E. coli. So then we generated a sort of pool, or actually not a pool. We generated 28 separate tubes of E. coli and added to each tube first one of these probes, then a second, so that each tube had a unique binary combination. And we allowed the probes to hybridize and then did all the appropriate washing. Then we combined all of the tubes into one pool and spotted this on a slide for imaging. And here's an example image of this mixture. So this is an artificial community of E. coli, 28 different sort of types of E. coli. And so we get an image that looks like this, although you'd be hard-pressed to identify 28 different colors, and there are a lot of reasons for that. But we don't need to be able to distinguish the colors with our eyes because this approach is all computational. So we also wrote software that can go through the image, identify the cells, and identify the specific binary combinations of fluorescent reporters that are in each cell. And that allows us to identify all of the 28 different kinds of E. coli that we had in that mixture. The next step that we did was to prove to sort of have a biological proof of principle of our assay. And so to do this, we identified 15 of the most abundant genera of microbes in the oral microbiome. And then we designed genus-level probes for these 15 organisms. We then acquired representative species of each of these genera from ATCC, grew them up in the lab, and made another artificial mixture of these 15 species. Then we applied our genus-specific probes to this mixture. And again, we're able to label all the cells in the mixture. We're able to use our computational tools to identify the cells in terms of now not just their fluorophores, but actually what the taxon of the microbes are. Okay, so then the last step was to actually apply this technology to a real oral microbiological community. So this is our fancy clinical protocol. We had volunteers donate floss. So we gave them floss, we had them floss their teeth, then they gave us the floss, and we took the floss with the attached biofilm and fixed the cells, put them in tubes. We sort of gently removed the biofilm from the floss and added our fish probes, and then spotted this natural community onto slides for imaging. And so this is an example of what we see, and I call this semi-dispersed human dental plaque. So what we see are clusters of bacteria and some individual cells. So we hypothesized that these clusters might actually be representative of what was there in the intact biofilm before we did our mild disruption. And again, we're able to identify the different genera of organisms in the community, and we scan through our images, and we see examples of multi-genus clusters of organisms. So then, because we have sort of disrupted this plaque, we didn't know if these were actually biologically relevant. So to try to get at that, we made model images of bacteria that were randomly dispersed on fields of view from the microscope. And then we were able to compare what we saw, the frequency of interactions that we saw in the models to what we actually saw in our samples. And that allowed us to build a network diagram like this. So in this diagram, each of these spheres represents a genus of microbe that we had in our samples. The size of the sphere represents the abundance of the organism, and a line connecting two of these spheres means that these organisms were found to co-localize, to aggregate together more frequently than we saw in the random models. Okay, so that's what I did in my graduate work, and now the members of my former lab have continued this line of questioning, and so they've improved on this now by not disrupting the plaque. So here is a sample of plaque that was removed with a pick and not disrupted. So again, what they see here now are these very large structures that are present in the plaque, and they actually give these a name. They call them hedgehogs. So here's an example of one of these structures. What they see is that these structures are sort of anchored by these very long filamentous bacteria, which stain with the probe for Corynebacteria bacteria. There are other filaments at different parts of this structure. These are fusobacteria and leptotrichia. And then at the ends of these structures, sort of like the cortex of this thing, are other organisms, mostly streptococci, some organisms of Haemophilus or Aggregatibacter, and others. Oh, and an interesting thing here is that these structures have been seen before decades ago in electron microscopy, and they're called corncobs. So what the group sees now is that these corncobs are actually parts of a larger structure, which they're calling the hedgehog. Oh, and also that they can be very diverse. So again, with the electron microscope image, you see a rod and cocci, or a filament and cocci, but you don't know what they are. With fluorescence imaging, now you're able to identify them at some level of taxonomy. So this allowed the group to sort of propose their own hypothesis, their own hypothetical model for hedgehog structure, at least. And this model is driven by environmental influences, host factors, and by the metabolism of the microbes themselves. So you have these long filaments, like I said, which are anaerobes, that attach to the base of the tooth. Then at the sort of tips of these filaments, you have bacteria that you have aerobes here that very quickly sequester oxygen and then produce an anoxic environment where the other filaments and other organisms live. So it's important that we think of these structures as ecosystems, because the fact that these organisms are interacting has something to do with their metabolism. So there are trophic webs that exist at all levels of structure in the human oral microbiome, and there is an influence of the environment. Although something that I've noticed is that more than sort of a macro scale biological ecosystem, or in addition to that, these structures also sort of look like an organism or an organ, right? Where you have a multicellular structure. You have cells that are making direct cell-to-cell contact. There's a repeatable pattern of development of these structures, but in contrast to a real organ, these are genetically distinct cells. So I close my short talk here with what I would propose as a framework for oral microbiome functional study. So systems level imaging of oral microbiomes is one part of this, one task that we have to do in order to understand these communities, their role in health and disease. This systems level imaging, of course, comes out of the molecular surveys that are concurrently being done. So together, these DNA sequence surveys and imaging allows us to sort of design these models of microbial co-occurrence and co-exclusion. In addition, some emerging technologies like metatranscriptomics, as well as metabolomics, are necessary to give a better understanding of these communities and to better inform our models. At the same time, as these exploratory surveys, I think we need these controlled laboratory experiments. Examples of these are growing biofilms in the lab based on hypotheses that we have generated from this data, and also computational modeling of the oral microbiomes. Taken together, both sides of these, both kinds of this science can come together and allow us to form testable hypotheses regarding microbial ecosystem structure and function. And with that, I thank all of my colleagues throughout the years and collaborators, especially the people that gave me space and intellectual guidance to do these studies. And also you for your attention. Thanks.
Thank you very much, Alex. So I trust here.
You mentioned how over time some of the microbiome constituent populations change in proportion as it goes from a healthy to a diseased state. Have there been any indications of ability to do chemical interruptions of this change proportions, like putting in some fluids like carbamide peroxide or something which would interrupt that anaerobic to aerobic chain?
Mm-hmm.
Yeah.
Here.
Well, you know, you use mouthwash and things like this, and these kinds of simple interventions can kill biofilms as a whole. But there really isn't a directed approach to work on particular groups of organisms within the biofilm, other than being able to address groups of organisms with antibiotics. And that's really about it. And as Niki mentioned, as far as treatment goes, we're still sort of in the scraping and cleanup area. So it's sort of limited, and there's really not a lot of possibilities for chemical intervention as you suggested.
So this may sound facetious, but should we be gargling with yogurt with lactobacillus? And it seems to be an unrepresented species, which in the intestine has some remarkable effects in displacing various toxic bacteria. Is this
well, it doesn't grow very well in the mouth. You know, you need to have organisms that most oral bacteria are really and truly found almost exclusively in the mouth. There are some that are found in the gut, and then there are these ones that Niki mentioned that somehow get translocated and become a pathogen. You know, but the oral environment is very unique and supports a specific subset of organisms, and it's difficult to introduce organisms into that group. That's a sort of a general observation from microbial ecology in toto.
But I just wanted to intercept to say that I think that Wynn's question is a really good one. Lactobacillus might not be the probiotic for us, but how can you shift the balance where you reintroduce what would be good to keep a healthy community and keep it that way and not let it shift is a concept that makes sense for the mouth as well. It's just that we don't know how we antagonize the shift yet. And for us, it might not be lactobacillus because that's not our healthy associated microbe that makes sense to keep the structure in a healthy state. But the concept could still of replacing with healthy bacteria still holds and is being looked at, I think, by many people.
I was interested with this interplay of obligate aerobes like actinobacteria to anaerobes to obligate anaerobes like Porphyromonas and Clostridia. And talking about this, when you use fluoride, it mostly kills anaerobes, right? That rely on glycolysis and not on Krebs cycle. So could you comment on the which is more important at which stage?
Well, early on in colonization, it's primarily streptococci that are the initial colonizers. And so these are facultative organisms and are very versatile. Later, these gram-negative obligate anaerobes come into the picture. But, that sort of understanding, as you saw from Niki's talk and mine, all of these organisms are there from the beginning. And so it's sort of a delicate balance. And if you intervene with one particular group of organisms, then the other group of organisms can come up. And so it's something where it's very difficult to intervene in a targeted manner. Because the organisms work also in a functional sense. In other words, you may be able to eliminate organism A from the community, but organism B, which has similar function, will then explode. It's too complex. The major problem, I think, for people with a medical background is to understand that this community is the pathogen. It's not one organism where you can just go in there and eliminate that organism and now the disease goes away. It simply doesn't work like that in these polymicrobial diseases like periodontitis and certain viral infections.
So,
GI
Alex,
tract.
can you answer what happens if you examine the floss after someone's brushed?
Yeah, so actually, you don't see anything. So our clinical protocol was actually our volunteers did not brush their teeth for 48 hours. That was the longest the IRB would let us do that.
Hopefully you do this experiment every day, at least once a day. You remove your oral biofilm from your tooth surface, but then it reappears and recolonizes, and it comes from other spots in the oral cavity. I like to think of the oral cavity as a big substratum-based fermenter, where you have saliva going through. Only the organisms that are adhered will be there. Everything else is swept out. So there's a large number of organisms in saliva, but they're on their way to the Mississippi River or someplace. They're not going to be important to oral diseases.
Mississippi River.
Potomac River, excuse me.
I had a question. Has there any work been done with salivary implants? Healthy salivary implants to a diseased mouth that would more stabilize?
I don't know that we're not there yet. So
The podium.
Okay.
So the question is, if I understand it correctly, is if we would transplant salivary glands. Now, the thing is that we are not saying that, or we don't know. No. So change of saliva, no, that has not been done. I know that there are studies where they've put saliva from different donors in mice and looked at colonization, looked at reversal or induction of GI disease, and I've seen that more in presentations than I've seen it published yet. But no, there's no transfer of saliva yet been done as an approach to change the microbiome in a host. And I don't know that that's been looked at this point.
I wonder, are all biofilms composed of microorganisms? When they speak of biofilm formation.
No. Well, biofilm, that's sort of a generic term, which it has various definitions. So I'll give you the classical definition. These are organisms attached to surfaces. That surface might be a rock in a stream. It might be a tooth in your mouth. It might be the inside of your cheek. But the concept is that they're adhered to surfaces. There's another concept, this is sort of my concept, that a biofilm is any place where organisms can interact. So any place where there's an interface. For example, you have have in lakes in particular, you have this pellicle that develops on the lake at the air-water interface, and this is a place for organisms to interact, and so I consider that a biofilm. In wastewater reactors, you have these suspended flocks, and these are organisms that are interacting together in the same time and space, and so that I consider also to be a biofilm. But they're not limited to bacteria. There's all sorts of organisms in biofilms.
Okay. Well, do we have any other questions? Yes, last one. Here we are.
So this study on the plaque, were your volunteers sampled in the morning when their mouth was closed for a long duration, let's say 7-8 hours right away, or were they sampled even though they may not have brushed for 48 hours, but was they sampled when their mouth was open and they were aerobic? What I'm trying to get at is the data that you're showing regarding anaerobic microbes and aerobic microbes, would that be different if somehow you could sample when their mouth was closed for a long period of time, where you could have significant amount of anaerobic buildup, which we know very well, which gives fair amount of toxic vapors. If you get up in the morning, you can, just for most people.
Well, the answer is we didn't control for that. So our volunteers actually had to come into the lab. So they came in whenever they came in and floss their teeth in the lab with us. So we did not control for that.
Does toothpaste help?
Very difficult question. So toothpaste is largely used for fluoride as well, and that is more to prevent from caries, not, and that, so that's against that, but also for the abrasive properties of being able to remove the biofilm. So in that sense, they do.
All right.
I'll add one thing to that, that there are toothpastes that are built up around remineralizing tooth surfaces that have been demineralized in a caries process. So this is an important health factor, is being able to remineralize these surfaces.
Okay, well, I want to thank you very much for a very stimulating discussion. Thank you all.